Hi, everyone, and thanks for joining me for episode 756 of People Behind the Science. Today, we are excited to be resharing our interview with our guest, Dr. Maureen Murphy, originally released on August 3rd of 2015. Listeners, if you have not met Maureen yet, she is an accomplished cancer researcher who is investigating the P53 Tumor Suppressor protein, which is responsible for stopping tumors from forming. Through her research, Maureen aims to develop more effective cures for cancer. And in our conversation, she talks more about her life and her research. So I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. Today, I am excited to introduce you all to Dr. Maureen Murphy. So, Maureen, thank you so much for joining us today on our show. How are you?
I'm doing well. Thank you, Marie.
Wonderful to hear. Well, I'm excited to introduce you to our listeners. So, listeners, just a little bit of background about Maureen. She is a professor and program leader in the Molecular and Cellular Oncogenesis program of the Wistar Institute Cancer Center in Philadelphia. She is also the Associate Vice President for Faculty Affairs and Associate Director for Education and Career Development there. Now, Maureen received her bachelor's degree in science and biochemistry from Rutgers University and her Ph.D. in molecular biology from the University of Pennsylvania School of Medicine. After graduate school, Maureen completed postdoctoral research at Princeton University, and she served on the faculty at the Fox Chase Cancer Center before accepting her current position at the Wistar Institute. And now, listeners, Maureen is here with us today to tell us about her journey through life and science. So, Maureen, we want to get to know you today. We are going to ask a ton of questions, but we want to get to know you both as a scientist, but also as a person. So can you start off this interview by just telling us a little bit about your life outside of science and what you like to do?
What I like to do outside of science is also devoted to nature. I like hiking with my dogs and traveling. Up until recently, my greatest love was running. I love to run 2 or 3 half marathons a year, but I have finally destroyed my knee. So I'm in the midst of recreating myself as someone who likes cycling. I've almost convinced myself that I like bicycling and yoga. So that's basically me in a nutshell.
Well, wonderful to hear more about life outside of science. I'm very impressed with your running. I must say, I am struggling to make it through 5Ks myself. So
you'll get there.
Now, tell us a little bit about these dogs. You mentioned you like to go out and enjoy nature with your dogs. What kind do you have?
I just lost my first 2 dogs, which was harder than eating barbed wire. I had a Shepherd Terrier and a Jack Russell, both of whom passed when they were about 13 or 14. And I just got a very unusual, so they're all rescue dogs. You get what you get.
Right.
This one is called a treeing Tennessee brindle. He's a coonhound. He's a hunting dog, full of energy, full of life, and that's the joy of dogs. I think when your life is so chaotic and everything, you forget to look at the little things and you forget to stop and smell the roses. Dogs always remind you to stop and smell this bush and look at this. It's fascinating. So that's why I love them.
Well, wonderful to hear about your family and the dogs that are included within them. I know myself have a dog, and it's the same thing. Every morning when I'm rushing around in a chaotic mood, the dog reminds me to stop, take a moment to pet her, and then quietly go out the door. Well, wonderful to hear about life outside of science, but I want to chat about your science next. So, Maureen, can you tell us how you describe what you do at work to someone who might not be familiar with science or your field?
I'm happy to. First, let me describe the Wistar Institute. I love being here. It's a small institute. It's the first biomedical research institute in America that's devoted to biomedical research, so it's the oldest in the country. It's just 30 or so scientists, and we just do science. But it's very disease-oriented research. We have great focus on cancer and vaccines, HIV, virology, things like that. And it's the 30 smartest people I've ever met in my life. My particular research is cancer research. I have wanted to cure cancer since I was 12, and that's usually what I tell people when I first meet them. So I work on the P53 Tumor Suppressor, which is the most important protein in your body. It's the protein that's responsible for first sensing the development of a tumor cell, a cell that's dividing when it's not supposed to be. P53 becomes activated and alarmed, and it tells that cell it's supposed to commit suicide or programmed cell death. Most tumors, as you might imagine, get around P53. So they mutate it or degrade it or get rid of it in some way. And it's almost true that a tumor almost can't exist without finding out how it can contain P53.
Well, so cool to hear about your research and wonderful to hear that you have been driven to do this since you were a kid. I love it. So now, can you tell us about motivation? I know a lot of scientists have little motivational phrases or quotes or sayings that they like to keep in their mind as they go through their day, something that keeps them going when times are tough. Do you have a favorite yourself, Maureen?
I do. I have a bunch of favorites, but my absolute favorite really spoke to me. So I learned this when I was in grad school. I was doing science and I enjoyed it, but I didn't know when was I going to fall in love with it? When was I going to be absolutely obsessed with it? And one of my favorite quotes is, to be successful, first fall in love with your work. And what it's really telling you is your work is never going to say, look at me, I'm beautiful. You have to decide to fall in love with it. You have to say, this work, however ugly it is, however whatever it is, I love it. And you sort of decide to love it. And I think that that's true in science. You have to program yourself to love it.
Absolutely. I really like that way of thinking about it. And I think you're right. With your work, it is sort of like a marriage. You know, there's going to be ups, there's going to be downs, there's going to be tough times, but you have to love it to get through it. Wonderful analogy.
Thank you. And just like a marriage, it's going to be a relationship that has its ups and downs, but you have to constantly put work into it to keep going.
Absolutely. Well, a wonderful motivational quote there. And I'm curious to hear about some of these people who may have motivated you along your journey. I know scientists get to work with so many inspiring people along the way. Can you share with us who some of your role models are?
Like most scientists, I always look for role models. I always look for people. And I cannot say, Marie, that I've ever found one person that truly embodied everything. Instead, I've found a bunch of people that I've taken bits and pieces from. One of my favorite people in science in the whole world, actually, is a scientist named Erica Galemis. She's at the Fox Chase Cancer Center. She's just the embodiment of not only an absolutely fabulous scientist, but a fabulous person, a very kind and giving person, always was there for any advice that I ever needed. Kathy Scotto, she's a dean at Rutgers University. You should absolutely do this program on Kathleen Scotto. She's fascinating.
Awesome. I've written her name down.
Oh, good. She's absolutely fascinating and inspiring person. All of these people are. Interestingly, the head of my thesis committee as a graduate student, that's supposed to be someone that you're not very fond of, right?
Right, especially by the end of it.
Well, is a guy named Dr. Stephen Liebhaber, who's over at Penn. He's right across the street. And now, 20 years later, I actually feel like his friend, and I've just always respected how he dealt with people. One of my other favorite quotes is from a scientist named Art Pardee. Now, he was a cancer researcher who's famous for discovering the restriction point. That's the point at which cells are trying to decide should they divide. It doesn't matter what you do when they get to the restriction point, they still divide. It doesn't matter if there's growth factors around or oxygen around, they get to that point and boom, they're going to go. Well, Art Pardee wrote a review for the journal Cell Cycle. And in that review, he said the best advice he got was from his grandfather, and that is that kindness is free. Kindness costs you nothing, and you will never regret it. And so that's something that really spoke to me. And in fact, whenever I bring someone into the lab, I make them read that review to teach them the importance of kindness. And the role models I've had are all people that seem to have known that already. Erica Galemis, Kathy Scotto, Steve Liebhaber already knew that you will never regret being kind to another human being.
Well, that is a wonderful story, Maureen. I think it takes me back to that book, like everything you really need to know you learned in kindergarten. It all comes back to things like kindness, some of these easy lessons.
Yes.
Well, wonderful to hear about some of these people who have had a big impact on you along the way. And I'm curious, now you mentioned the Wistar Institute is a relatively small institute with only about 30 scientists. So do you find yourself collaborating a lot with some of these people at different institutions, or is it fairly well contained?
Wistar is different from any other place I've ever been. So I've been at Penn and Princeton and Fox Chase and Wistar now. This is the most collaborative group of people that you could ever imagine. We always have these grants from the National Cancer Institute that help run your facility. They run the core facilities, the microscopy, the electron microscopy, proteomics facilities. And these are very important grants to get. And one of the things the reviewers of these grants look at is how collaborative are your people. And the benchmark for collaboration is they like to see 10% of all of your publications being collaborative. And at Wistar, it's over 50%.
Oh, wow.
Yeah, so it's just this group of people where the culture is, you bang on someone's door and say, this is what we must do. So it's been thrilling to be here. It's just been incredible. We are at the heart, and I think we're the heartbeat of the University of Pennsylvania. So the Wistar Institute is right in the center of Penn's campus. A famous quote about P53, the tumor suppressor, is, no matter which way cancer research turns, P53 comes into view. It has so many roles in inherent aspects of cancer, and it doesn't matter where my research goes. There's either an expert here at Wistar or there's an expert across the street at Penn. So it's very energizing to be on this campus. Everywhere my research goes, there's someone who's absolutely the world's expert in that pathway, either here or across the street.
Well, wonderful to hear that you get to work so collaboratively, and I think so exciting to be pursuing this important endeavor of finding a cure for cancer. And you mentioned this is something you've been passionate about since you were 12 years old. So Maureen, I want you to take us back to the beginning and tell us where this passion started. Can you share one of these earliest memories that you have of being interested in science or cancer research?
Sure, Marie, but I'll tell you right away, this is an embarrassing story.
I'll try not to laugh too loudly.
When I was about 12, I realized that I seemed to have a lot more energy than other kids. I just always felt like I had this seething energy inside of me. I felt like I knew the answers to the questions the teacher was going to ask even before they were done asking it. I just realized that very young, I was going to have to rein in this passion. I was going to have to rein in all of this energy and choose an avocation that was going to suck it out of me, that I would really be able to throw my entire body and soul into. And the first choice that I made was to be a nun. I was an Irish Catholic girl and went to Catholic school, and I thought of Ingrid Bergman in, what's the movie, The Bells of St. Mary.
Oh, sure, yeah.
And I thought, that is going to be me. I'm going to be a nun, and I'm going to teach the little kid how to box and things like that. And that lasted for 1 week.
Really? Wow.
I would say, well, when I'm a nun, I'll be doing this, and when I'm a nun, I'll be doing that. And after a week, I thought, that's just not me. There's nothing wrong with it, but I see things that I don't care about. I didn't care about how nuns were always sort of a little subservient to men, and I just thought, not me. What am I going to choose now? And the second thing I chose was curing cancer. I realized that this was going to be a problem that I could devote my entire life to. I could work 80 hours a week after and maybe not even get a cure, but at least be able to advance a cure that it was going to absolutely suck every ounce of energy I have. And Marie, as you know, as a fellow scientist, science will take everything out of you.
It absolutely will. So this is a fairly big jump then in terms of your career aspirations. Had you had an underlying interest in biology, biomedical things before that, or was this just out of the blue something that popped up that you then kind of latched onto and became very passionate about?
Out of the blue, I latched onto it. I did fine in science as a kid. And when I went to school, I went to Rutgers, and I was much more interested in literature. I wanted to be a writer. And so I decided to be a dual major, English and biology. And about junior year, it was exhausting. It was just too hard to maintain two majors. And I took a class called molecular biology, which of course 25 years ago was brand new, and you were looking at the absolute inner workings of the cell. How does the DNA go to RNA, go to protein? And I was absolutely hooked. As soon as I took that molecular biology course, Alice Wu was my professor. Still remember her. That was it. I'm going to go to graduate school and do molecular biology. I think it was great that I had literature as a side passion, because most people don't realize in science you have to be a good writer. If you are not a good writer or communicator, you're not going to do that well. You have to get your word out there, and you have to write papers and grants. And so it's helped me tremendously.
Certainly. You brought up a really good point. I think this is one of the things that a lot of scientists struggle with is they hate writing. So it is drudgery to write those papers, those abstracts, things like that. So great that you had that dual passion there.
Here at Wistar, I actually teach writing, and I teach a 6-week, very writing-intensive course. And I can just watch people as it occurs to them, oh, that's how I want to communicate my thoughts, and that's how I can make it easy for the reader, and that's how I can sway and persuade. I think it's so, so important, and a lot of times it's ignored.
Absolutely. I think there's definitely an art to scientific writing, which is a little bit different from other forms of writing. But you're right. There's a lot of persuasion. There's a lot of finding exactly the right words to convey it within a small space. So.
Right.
Wonderful to hear about your career path to get you to where you are today. And we kind of got you up to graduate school. I know you spent some time, like we said, at the University of Pennsylvania, and then you did your postdoc at Princeton and then went to Fox Chase Cancer Center. So can you tell us a little bit about how you flowed between those positions?
Penn, it's an interesting story. When I started out in science and I was this young punk graduate student and I said, Steve Jobs, I'm going to bend the universe. And I decided I'm going to clone a new tumor suppressor. And I was interested in brain tumors. They're so devastating. And I was able to culture some brain tumor cell lines. And I decided I need normal brain tissue. And I'm going to make a normal brain cell line. And I'm going to do this technique called subtractive hybridization that will allow me to know what genes are expressed in the normal brain cells and not in the tumor. And I was able to, at a huge medical school, to get tissue from fresh autopsy and culture these brain cells, which are growing to this day and no one else has ever succeeded at, and then did subtractive hybridization. And I thought, I'm going to clone this tumor suppressor. And I cloned several genes, which now turn out to be very important in cancer. One of them was an IGF, insulin-like growth factor binding protein, that now is known to be downregulated in most tumors, and it's very important for tumorigenesis. But when I was looking at this, IGF-BP7, I wanted a gene that was a tumor suppressor, so it should be deleted in human brain tumors. It should be rearranged or deleted. And I was doing these southern blots to look at the structure of this gene, and I saw what looked like deletions. And I was so excited. This is it. This is my cell paper. I'm going to be famous. And it turned out that it was artifactual.
Oh, no.
I had a contaminant in my probe, and it was picking up another gene, and it was complete artifact. That was probably the only time in my career that I was just devastated. I thought I had this cell paper, and now it's just nothing. And what am I going to do? And what I decided, I think, is an important lesson for other scientists. And that is that you should not be in this for the glamour. That's the wrong thing to be in science for. And you should not even be in it to prove your hypothesis. That's wrong too. You should just be in it because you're in love with the question you're asking and the potential that the answer can help humanity. And if that's your motivator, then you can survive the ups and downs that we all have to go through much better. I'm in love with this question and the potential that it will help humanity, and that's all I can see. I'm not in love with fame. 3 scientists a year get famous.
Right.
I'm not in love with glamour. Most scientists are not glamours. They're geeky like I am. And I'm not in love with anything like that. I'm in love with the question. And as long as you are, you can survive funding crises, which, Marie, there's a terrific funding crisis right now.
Definitely.
You can survive anything.
Well, Maureen, I love that this theme keeps coming back about how passionate as a scientist you have to be about your work. And I think this is something that a lot of scientists have in common is they are so excited to go in every day to do their work and really have the potential to impact the world like you're describing. So great to hear about your experiences. And now I want to know a little bit more about what you're working on today, Maureen. We mentioned some of the work that you've done over the course of your career, but can you tell us about a project that is going on right now that you are so thrilled to be a part of?
I have 2 projects right now that I'm really thrilled by. I try and run my research laboratory so that I'm doing very basic science where what we find has the potential to help people now, but more potential to help people in the future, and also very translational science. So in other words, how can we help people who are being treated for cancer right now? So I have a basic research component that is devoted to the P53 tumor suppressor. How does this protein kill a tumor cell? And then I have a translational component, and that is on a new drug that we have. And that drug is an inhibitor of a protein called heat shock protein 70, HSP70. HSP70 is a great tumor target. It's a protein that's overexpressed in 70% of all human tumors, and it's almost undetectable in normal cells.
Wow.
It's a stress-induced protein, and it helps the cell survive stress. And tumor cells are under terrific stress, and they really need HSP70 to help them survive. So I call it the Achilles heel of tumors, that the reliance of tumors on heat shock proteins to help them survive is an Achilles heel because normal cells don't need these. So we have an inhibitor of HSP70. Now, when you have a drug, It's thrilling. You add it to tumor cells, they die, you give it to mice, and the cancer is cured. But they call it, you're in the valley of death. Trying to get that drug into humans now costs millions of dollars. It's almost impossible for a small research lab like mine to get it there. And what you really have to do is have a very strong, powerful, and experienced collaborator. And we were very lucky. Last year, we entered into an academic collaboration with GlaxoSmithKline to develop our HSP70 inhibitors. And that's been unbelievable. The P53 work that we're doing, I'm equally excited about because of the following reason. Most people don't know this. We know that there is genetic variance in everything. You have blonde hair, I have brown hair. You have blue eyes, I have brown eyes. And that doesn't necessarily mean anything for cancer, but P53 has genetic variance. You and I differ in our P53. Most people don't know that.
Interesting. I didn't know that either, yeah.
And when you talk about most genes that are important in cancer, there aren't variants. Those genes are so important that there's not genetic variance. But P53 does have genetic variance, and I was fascinated by this. And I was more fascinated by the fact that 2 of the P53 variants are more common in African Americans. African American and Hispanic Americans tend to have a particular form of P53 that's different. And what we found in the lab is not only is it different, it's actually less effective as a tumor suppressor. So it doesn't kill tumor cells as well. And so I sort of said, African Americans have the largest burden of cancer in the United States. They're more likely to get cancer. They're more likely to progress. They're less likely to be treated well. And it's been known for years that that component is genetic. If you equalize everything, the same stage of tumor, the same treatment, the same everything, African American women are more likely to die from breast cancer than Caucasian American women. It's a fact. And so what we ended up finding about this P53 variant, we created mice that have this variant of P53 versus what we call wild type. The wild type variant is the most common variant. It's not like one is bad or one is good. The most common is wild type. And then we created mice that have this African American variant. And long story short, the mice started getting cancer, and we were shocked. And then we did some population studies, and sure enough, this variant explains part of the reason why African American women more commonly get more aggressive breast cancer. That work has been submitted to the journal Cancer Cell and is currently under revision. We just sent it back yesterday, so knock on wood, Marie.
Oh, wow.
Send me your good karma.
Right. We'll keep you in our thoughts, yes. Well, this is really fascinating work. So is there anything we can do with this knowledge? Is there anything we can do to particularly help certain groups who might have a greater susceptibility to cancer because of P53 variations?
That's a great question, Marie. So I think the reason I'm so passionate about this variant and giving increased risk is, for the most part, minority health disparities have largely been ignored. And so I feel like we have the potential to really open up. So I tell people it's somewhat satisfying to find a genetic variant that might predispose to cancer and tell people, you need to be more cautious, you need to screen more, you are at risk. That's somewhat satisfying. We also find, so one of P53's jobs, in addition to killing a tumor cell, is responding to chemotherapy. So its job in a tumor where it's there is chemotherapy or radiation therapy signals to P53, and that's how it kills. And we found a couple of chemotherapeutic agents that don't signal at all to this variant. Well, that's somewhat satisfying because we would tell a person, you are at increased risk, and these 2 drugs you should never get if you have cancer.
Sure.
Somewhat satisfying. What we've done now, Marie, is we have a screen for what drugs kill those cells better. And we just found the first 2 drugs that actually kill cells with this variant better than they kill cells with wild type P53. So now we can actually personalize medicine. We can tailor chemotherapy to who this person is, and that's exciting. So that, to me, is the future of this research, to not only inform who is at cancer risk, and not only inform, oh, you should not get these drugs, but to say, this is how you should be treated. This is how your P53 will respond best.
Well, that is phenomenal, Maureen. I really like this idea of personalizing medicine and specifically treatments to cancer based on what we know about people's genetics. I think this is just incredible research. So very exciting to be a part of it. And I know we mentioned some of the struggles that you've encountered along the way to get to this point. You mentioned this one moment in your career when you were thinking back on these contaminated results and being so disappointed by them. And I think challenges, roadblocks, things like these happen a lot in science. You get these failures. So can you think back now on your career and tell us about another instance where you really struggled with something or you were really disappointed by a failure? Because I think people don't realize that this happens more often than not, I think, in science.
In thinking about this program with you, Marie, the one thing I wanted to bring up in this program, probably the only thing that I wanted to make sure I said, is exactly how difficult it is right now to get funding for research. In any trend, the stock market or whatever, there are recalibrations and readjustments. And so there have been times in history where a ton of research money has been funded towards cancer research. And for the last 10 or 15 years, we've gone the opposite, where Congress has failed to increase budgets, where instead they're decreasing budgets, and with cost of living increases, we're getting even less money. And now is probably the tightest time in the history of United States science to get funding from the National Institutes of Health. I would say funding rates at the National Cancer Institute are 9%. Well, what does that mean? That means 91% of people write grants and don't get them.
Right.
If you knew you had a 9% chance of success for anything, you would probably think twice about what you're doing. And that's okay. So we should fund the 10% of the best ideas, right? That's okay that it's hard to get research funding because we should be picky. But it's been 10 years of that, and that's hard to survive. And what I see from my friends who are scientists, who are fabulous scientists, they're starting to drop out of science. And that is a criminal shame. For the first time, I see scientists who are passionate, who are doing absolutely cutting-edge research, but through bad luck or bad circumstance are not getting their grants and are leaving science. And I don't think the public is fully aware of really the crisis that we're having in biomedical science right now, with funding being so tight for so long. It's fine to make funding hard to get. It's fine to make scientists fight for it and try and do better, but it's been too long. It's now to the point where really good scientists are starting to leave.
Certainly. And as an investigator in the same process of trying to submit grants to the NIH, to a variety of different funding agencies, it is a struggle. I think you hit the nail on the head there, and it has been going on for a long time. And it is so challenging to be that person who might be in the 11th percent or the 12th percent, where, you know, you are so close, but you just can't get there. And I think this is something a lot of people are struggling with. So is this something currently right now that your lab is struggling with?
Well, knock on wood, one of the advantages to my coming to Wistar was there are a lot of grants here that are highly collaborative group grants. They're so-called program projects. That's where you bring 5 or 6 investigators together to compete for a large amount of money to put all of their brains towards one process. And when I first got here, I was funded okay, but still always very tight on money. But the head of Wistar, a guy who's brilliant named Dario Altieri, actually asked me, would I be part of this program project on melanoma, which I went, sure.
Yeah, sounds great.
I came to learn that the Wistar Institute is the absolute world's best place to work on melanoma. The resources here are unbelievable. The group of investigators here focused on melanoma are unbelievable, and we have clinical collaborators all over the world. And so through Dario's vision, I was placed on this program project. I took my HSP70 inhibitor and started targeting melanoma, and we're about to publish a beautiful paper that it's a great drug for melanoma. And so I was able to get money from the program project. Since then, I've gotten other funding from melanoma. My P53 research, knock on wood, is being reviewed this week and next week. I have 2 grants out. I really need 2 or 3 grants in order to fund my research. And knock on wood, it will do okay. But the truth is, I'm as scared as everybody else.
Certainly. Well, Maureen, it sounds like we caught you at a critical moment here. You've got a paper revision just sent in. You've got grants and reviews. You might have a lot to celebrate in the next couple of weeks.
Or not.
Definitely. Well, wonderful to hear about some of these challenges. I think it is so important to help people become more aware of some of the challenges that scientists are facing, some of the struggles along the career path that perhaps young scientists will have to face if they're interested in this journey. But we don't just want to talk about the difficult periods, the challenges, the struggles, the failures. I also like talking about successes. And we've talked about some of your successful research projects. But Maureen, I want you to tell us another success story, one that has really meant a lot to you. It can be even just a small victory, like mentoring students or things like that.
I have to say this, a big transition for me when I started my career was I going to do research on animals? I didn't know how I felt about doing research on mice. I certainly didn't want to have any mice die. And I really struggled. And what I decided, to be honest, is you can do anything you want in a test tube or even in a cell in a plastic dish. It's not necessarily real. But if something happens in a mouse, it's real. If you cure a mouse of cancer, that's real. It's a lot more real than just killing a cell on a dish. So I decided to transition to making mouse models of P53 variants. And that's actually been thrilling because the biology is automatically so much more complex. And we've ended up making mouse models of all of the P53 variants there are. Now, one of them is a particular variant at amino acid 72. And we had this hypothesis that it would have this big role in cancer. But it turned out it didn't. And a lot of genome-based studies were showing that this variant had a big role in diabetes, and we're about to submit a big paper on diabetes. Now, that's where being at Wistar, where there were diabetes experts, and there were certainly diabetes experts over at Penn, really helped. So I think that turning to animal models and getting over my fear. It was a friend of mine, to be honest, who said, the American Indians, when they were going to kill a buffalo, said to the buffalo, listen, we're only going to kill enough of you to survive. We're not going to kill more than we need. And do you give yourself up for the betterment of humanity? And apparently the buffalo always said, sure. Right. And my mice have always said, okay. But that's been a big transition. You brought up training. I love training. The beauty of science is how much exposure to young people you have. It keeps you young. There's always new people in the lab who are young, who are just starting out. We have high school students, college students. We have a big program here at Wistar called the Biomedical Technician Training Program, where they teach young students who are getting their associate's degree to become a biomedical technician. So there were always young people here, and I love it. There was people that when I was young gave me a shot, and I love giving them a shot and seeing how they then go on and prosper.
Definitely. Now, we mentioned in our introduction that you are also the Associate Director for Education and Career Development there at the Wistar Institute. So is this part of your job there? Do you work with students there, and is it development for students, or is it more on the faculty level?
It's both. So one, I run the training program here. So for people who are graduate students as well as postdoctoral trainees. And the training program I've created is a lot of writing. I make them do writing, because I tell them if you do good writing, you will do well no matter what.
Right.
But also enable them to pick their career path. As you know, science now, it means many things. It means science writing, it means communication, it means being a patent lawyer, it means teaching, it means so many different things. And so the training program we've created here at Wistar accommodates all of those things. We actually have teaching workshops where we bring people who teach for a living to tell us about how to write a syllabus, how to write an exam, how to get your lectures to be really exciting. What are new apps that kids will use that will help them learn and retain information, which is actually really fascinating. We have career development seminars. I also mentor the junior faculty here. It's a tough time to be a junior scientist with funding so low. So the junior faculty mentoring program is going through their science, helping them write a grant, but also introducing them. This has been really big. I've brought in program officers from the NIH to tell them about new funding opportunities, the little things that you don't necessarily know, what's advice that they would give. I've brought in editors of journals to bring them in to say, what do you need to get your paper in my journal? And it also gives that editor one-on-one FaceTime with junior faculty so that they can say, oh, I remember that guy. I met that guy. Or I remember that woman. I met her at Wistar, and I remember her science. It was really neat. So just things to help people find out where they want to go and then get there.
Well, this sounds like a fantastic program. And I think these training opportunities on both levels that you're describing here, I mean, for graduate students, there are so many lessons that you don't learn in graduate school that you will need later in life. And one of these is teaching. You get to your first class as a junior faculty member and you have no idea how to make a syllabus, no idea how to go about making a whole semester's worth of lectures. So I think that is critical.
When I was applying for jobs, my first job, one of the questions I was asked is, what is your teaching philosophy? And I went, what? What is that? I remember writing something so ignorant, like, I'm a good communicator and I'm a good teacher and I care about it and that's my philosophy. And now I look back and cringe at that. So part of our teaching workshop here has been to tell people who are interested in teaching what is being looked for when they ask that question.
Well, so important. Glad to hear that you guys are dedicating your time and resources to helping develop the next generation of scientists and help develop the faculty that you currently have working with you. I think those are so important because these are critical times in people's science careers. So wonderful to hear about some of these success stories, Maureen. It sounds like you've had some wonderful times along your journey as a scientist. And I think one of the aspects that we talked about that doesn't get as much attention as I think it should in careers in science is opportunities to travel. We mentioned all the different places that you've gone for your training, but I think scientists also get to travel quite a bit for conferences, to work with collaborators, things like that. So can you think back now on all the places you've been for science, Maureen, and tell us about one of your favorite destinations?
It's true, scientists get a lot of opportunity to travel, and they're innately curious human beings. They want to see the world. They want to see inside a cell, but they want to see outside too. The P53 Tumor Suppressor is obviously the most important protein in your body. There's actually a meeting devoted to P53, and it's called the International P53 Workshop. And that meeting is held every 2 years by scientists. There's about 400 scientists in the P53 field. I hosted it 1 year here in Philadelphia, but it's hosted all over the world. And so this has accounted for the majority of my travel. So went to Greece, Scotland, China, Australia. And it's an interesting group of people because they acknowledge that the dynamics of interaction are just as important as the science. So there are many opportunities to go on trips together as scientists. So, for example, when the meeting was in Crete, we went to the ruins of Heraklion to see. But my absolute favorite trip was the P53 meeting in New Zealand. New Zealand is the most amazing place in God's green earth. It was just an unbelievably beautiful, beautiful country. The people were so friendly. It was easy to travel and just unbelievably beautiful. It's a tiny little island. It's actually a series of 2 islands with the most diverse geography. There's glaciers and rainforests and volcanoes all within 4 or 5 miles of each other. I went to a place that was a town in the center of a dormant volcano. You have to sort of drive up the volcano, then drive down the volcano, all on the opposite side of the road, of course. And sitting in the center of this dormant volcano, it had filled with salt water from the ocean. And dolphins had figured out how to swim through the caverns underneath the island into this now protected saltwater lake, just playing and frolicking. And I remember thinking, if I could get my dogs here, I would stay here the rest of my life.
Well, that is so cool. I went to New Zealand, but it's been quite a few years now. It is just like you're describing a beautiful country. So wonderful to hear about your experiences there, Maureen. And I think the opportunities to travel are phenomenal in science. And it's really cool to hear that you have a whole field dedicated to this really important P53 protein that you're studying. So very, very cool. And now we chatted a little bit about your life outside of science at the very beginning of our interview. And I know a lot of scientists, when they're able to escape from the lab for a little bit, perhaps take a break from reading the literature, they're able to read some fantastic books. And I have gotten some phenomenal recommendations from different people we've had on our show, Maureen. So I want to ask you today, do you have a favorite book that you've read that you would like to recommend to us?
My favorite book in the whole world, I think I've read it probably 4 or 5 times, is The Emperor of All Maladies. It's the history of cancer research. It's not just a fascinating history. It doesn't just focus a lot on the young people who lost their lives as they went to clinical trials, and then the young people who were cured of cancer, but it's also beautifully written. So I would highly recommend The Emperor of All Maladies. Right now I'm reading a much shorter book that everyone in the world should read, and this is Advice to a Young Poet. This is by Rainer Maria Rilke. Unbelievable. So he's an interesting person. He's a guy who was born in Czechoslovakia, a very frail, timid, shy, introspective poet. And because his parents split up in a divorce, which was devastating enough, they threw him into military school. So he's in his version of hell, figuring out, how do I stay true to myself? How do I get through this? How do I survive? And advice to a young poet is his series of letters that he wrote to a young man 15 or 20 years later who wrote to him from the same military school saying, I'm in hell. How did you manage this? How did you manage to get out? And it's just some of his advice is just wonderful. One of his quotes is something that everyone should follow. And he said, my advice to you is to go inside the depths of your soul and scrape the very bottom of your soul and use all of that life energy towards what it is you want to do, which, of course, when you're trudging towards the train at night or whatever, you maybe are not scraping the depths of your soul. It's something we have to be reminded to do. Am I really using everything I've got towards this question, or am I just sort of floating along? And so it's wonderful advice. I think young people today, a lot of them are in a situation where they feel a little bit of angst. I'm not where I want to be. How am I going to get there? And his advice is really, how do you stay focused on your journey and ignore all of the stuff around you that might be sort of dragging you down? It's absolutely fabulous. And it's much shorter than The Emperor of All Maladies, which is pretty long.
Gotcha. Well, this sounds like a phenomenal recommendation. And you brought up some really good points here about some of the struggles when you're in this period of being a young person, trying to figure out what you want to do with your life and how on earth you are going to get there. And I think a lot of our listeners might be in the same boat where they are perhaps interested in a career in science, but they're not really sure what the options are. They're not really sure how to get there, and they're not really sure if that job is going to be there. So this sounds like a great book that kind of traverses that gap for them.
And I think the 3 lessons we talked about today, Marie, are the most important. Force yourself to fall in love with your work. Scrape the absolute bottom of your soul. I think Steve Jobs said, we're here to bend the universe. So don't forget that you are able to bend the universe if you want to. And then finally, kindness has to come first, that you will never regret it. It's free. It costs you nothing. If you live your life by those 3, you'll do okay.
Absolutely. Well, I think these are excellent lessons to share with our listeners today, Maureen. And I know we've chatted throughout our interview about the different people that you've gotten to work with over the course of your career as a scientist. And I know there are stereotypes out there about what scientists are like. Some of them are more true than others. And one of the things we love to emphasize on our program is this human side of science, because scientists, we are so passionate about what we do. We're excited to go into work every day, but we're people too. We like to go out and have fun and have a life outside of science as well. So can you think back now on all the different groups you've worked with, all the different personalities and quirky traditions and things that you may have experienced? And can you share with us one of these funny human side of science kind of memories?
Sure. If my lab has a quirky tradition, it's one that we got from other labs. Science, when you're running a research lab, is transient. You have people in your lab for 3 to 4 to 5 years, and then they move on. So your scientific laboratory is a stepping stone. It's not the same group of people for 30 years. It's a constant flux of young people who then go on to other things, greater things. They go on to a postdoctoral fellowship or a job in industry or a job as faculty starting their own laboratory. And so our quirky thing is when someone leaves the lab, we get them that Dr. Seuss book, Oh, the Places You'll Go.
Oh, love that book.
It's such a great book. It's such a lovely message. And then we scribble through the whole book. This is you, and this is your battle against ovarian cancer, and this is this, and just sort of warm memories. And so we fill the book entirely with pictures and writing and memories and things like that. And there are actually now editions, because other people do this, where they have spaces for you to write things in. And so that's our lab tradition. When someone goes, we fill that book with just memories and advice and things like that. It's so much nicer than just a card, have a good day, good luck, goodbye. You're bringing all of those people with you wherever you go.
Well, I love this tradition. I love this book that you're able to put it all into so that someone can take it with them. Like you said, it's not quite as meaningful when you just have this card that you're probably going to recycle or throw away or whatever afterwards. So this is a great tradition. How did it get started?
It was a woman, Becky Raftianis Blanchard, was an investigator next door to me at Fox Chase, and it was started in her boss's lab, Dick Weinschelbob's lab, and she taught me it, and now it's definitely taken over. I've seen other labs at Wistar start to do this as well.
Well, I love it. And one of the things that I love about a lot of these traditions that you hear about in different scientific laboratories is that they are passed on not only through the generations within a laboratory, but oftentimes people who leave the lab then take them with them to wherever they go. So it's so cool to see them proliferate. So wonderful to hear about the human side of science from your experiences. And we've chatted a lot about the work that you've done over the course of the years and some of the research questions that you've been able to tackle. But I think every scientist has on their to-do list or their dream question list one of these big questions that they are just dying to know the answer to, but there might be something standing in the way. We mentioned things like funding that often get in the way, but also staff, technology, feasibility, things that might stand between you and being able to answer these questions. Maureen, if we took all of these out of the equation, is there one question that you are dying to answer?
I often think when you commit yourself to a field, and I've committed myself to understanding P53, what it does, what it means for cancer risk and cancer therapy. But if I was not in that field at all, to me, the most cutting edge question regarding cancer is where is the tumor? Where are micrometastases? So it would be in cancer imaging. Why don't we have better ways to see where a tumor is? Why is it so hard for us to see all of the little micrometastases that ultimately are what kills the cancer patient, not the primary tumor? So I think if I had $1 billion, I would try and make molecular probes that would actually track to a tumor, that would actually say, here it is, so that you could know that you eradicated it with chemo or radiation, or that you could know that it was there so that you could cut it out. I think one of the futures of cancer research will be in imaging technology, whole body imaging to say exactly where's the cancer. We always know where the primary tumor is. That's where we have the pain. That's where we have the lump, and that can get cut out. But we don't know where did that tumor go and sort of seed itself in the rest of the person. And it's those that we don't know where they are. And if we had imaging to say, here it is, then we could get better cure rates. And cure rates have gone up tremendously. But of course, we can always do more.
Well, this is a great question, and I'm really glad you brought it up because I think it's something that a lot of people outside the field of cancer biology might not even think about. I know myself, I'm always really focused on kind of killing the tumors, but right, you have to know where they are first. So, great point, Maureen, and wonderful question for us to think about. And throughout our interview today, we've talked about some great pieces of advice that you've picked up along your journey from various sources. And I want to know, is there a best piece of advice that you want to share with our listeners today?
One of the pieces of advice that I got from my old boss, Arnie Levine at Princeton, was when you start your lab, bear in mind that you're never going to get people to work for you who are as good as you are. And I thought, okay, that's good. At least I know that I'm going to get people that I really have to work with. I'm actually happy to say, Marie, that advice was wrong.
Really?
I actually get scientists who are better than me. And so it's my pleasure to mentor them. I end up learning just as much from them as they learn from me. So maybe that's true. The best piece of advice I got was actually wrong. I've actually been able to mentor people who are just as strong and in love with science as I was. And maybe that's a positive note to think about.
Wonderful piece of advice to share. Now, Maureen, is there any other closing note that you want to end on?
This is the advice I give everyone in my lab. I say, listen, your life is like a pie, and you have pieces of your pie that are labeled whatever you want to label them that make up your life. And maybe you have a big piece that's family because you're a family person, you're devoted, and that's great. And maybe you have a big, huge piece that's work, and that's great. And a piece that's fitness or finance, and you make those pieces how big you want them. I don't want to devote a lot of time to finance, but some. And I said, so always look at your pie and make sure that you're calibrating, that you're not spending too much time on any one of those things, that you're not ignoring one of those things. But the advice I give people, you make your pie, but make sure one piece of that pie is volunteering. Make sure that you're doing something. So my whole lab, we all run. And we all run the Susan G. Komen race, and we raise money for breast cancer research. And we all run the Broad Street Run in Philadelphia, a fabulous run, and we raise money for the Wistar Institute. So make sure one of your pieces is giving back to your community.
Absolutely. This is fantastic advice for everyone. I think it helps you because it is not only you're helping your community, not only you're helping your institution, but it's a rewarding thing that allows you to get together with different people, maybe outside of the lab in a slightly different context and feel like you're giving back. All this talk of pie is making me hungry, Maureen, I must say. Well, thank you so much for your time today. Can you tell our listeners now how they can reach out if they want to learn more about you and your work?
Absolutely, Marie. So you can learn more about the Wistar Institute at www.wistar.org. And you can actually find a link called Find a Scientist and find me, Maureen Murphy, or you can even email me. I'm at mmurphy@wistar.org.
Perfect. Well, listeners, definitely reach out, figure out what Maureen is working on, get all the details on the website. And Maureen, thank you so much for joining us today and sharing a piece of your story.
Thank you, Marie, for this wonderful program. I've loved hearing your other podcasts. It's been absolutely fascinating to listen to these other scientists.
Well, I appreciate it. And I appreciate you listeners as well for joining us. Thank you so much. And we'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.